So novel beta-lactamase inhibitors have become a critical drugs to combat the most resistant gram-negative infections of the 21st century, I would say. A series of new compounds with even more broad and potent activity are in the horizon to add to the therapeutic armamentarium. And I think these are exciting times when we have new drugs that potentially can treat our most recalcitrant gram-negative infections. Today, we will have outstanding world-class experts discussing these drugs. The objectives of this podcast is to review the beta-lactamase inhibitor combinations that are currently available in clinical practice. We're going to discuss future perspective of this armamentarium of beta-lactamase inhibitors that I'm going to call BLI, and provide insights into resistant development for this class of antibiotics. So this episode is brought to you by Antimicrobial Agents and Chemotherapy Journal, available at asm.org/journal/AAC. I am your host, Cesar Arias. I'm the editor-in-chief of AAC, and this podcast is supported by the American Society for Microbiology, which also publishes AAC. This is important. If you plan to publish in these top journals of AAC, like AAC, ASM members get up to 50% off in the publishing fees. Visit asm.org/membership to sign up. Don't forget to check as well our latest issue of AAC, available at asm.org/journal/AAC, with outstanding papers on mechanism of resistance, pharmacology of antimicrobial agents, epidemiology, and clinical therapeutics. I'm sure you will find a paper of these new beta-lactamase inhibitors in the current issue. Joining us to discuss these important topics are Dr. Robert Bonomo, who is professor at the Department of Medicine, Pharmacology, and Molecular Biology and Microbiology at Case Western Reserve University, and is director of the VA Care Center at Cleveland, Case Western and Cleveland VA, Ohio. Patricia Bradford, who is from Antimicrobial Development Specialist in New York, United States. Welcome to the program, and thank you for accepting my invitation. So we have a hot topic in front of us, so let's get into the conversation right now. Let's start with you, though, Patty. Tell me, explain the concept of beta-lactamase inhibition, briefly, and how that has been a very successful concept clinically.
Mm-hmm. Beta-lactamase inhibitors have tended to be mimics of beta-lactam. Either they're beta-lactam antibiotics in and of themselves, or they're mimics structurally, and they interact with beta-lactam antibiotics. They interact with the beta-lactamases, and the beta-lactamase is inactivated by this inhibitor. It either has a very long binding time, and then the turnover is very slow, and the molecule is then destroyed, or it's a covalent binding. But anyway, the point of the inhibitor is to take the beta-lactamases out of the equation for susceptibility and let the beta-lactam antibiotic do its job of binding to the PBPs.
And Robert, you said you've been using this for a long time, and so am I. And that seems to work clinically. And this concept of inhibiting one of the most important mechanisms of resistance in gram-negative seems to work. Would you agree with that?
Yes, it does. And I think you and I have, and many people in the clinical world have really reaped the benefits of this unique combination. And it allows you to keep drugs that normally would be susceptible to beta-lactamases in gram-negative bacteria. When you partner them with a beta-lactamase inhibitor, that potent beta-lactam is still around to do its work. And like Patty said, inhibit the enzyme by whatever mechanism, the beta-lactam can work its effect. And, how you choose the beta-lactam to partner with the beta-lactamase inhibitor, I've talked about this many times. To me, it's like having a good marriage. You got to pick the right partner, because if you don't pick the right partner, the beta-lactam is not going to function at its highest level, and there's going to be some compromise there. So it's and like I said, clinicians have taken advantage of Ampicillobactam, amoxicillin, clavulanic acid, Piptazo, and then we have our new beta-lactam, beta-lactamase inhibitors. So we've gotten a lot of clinical benefit from them, and they've still, as Dr. Bradford said, challenged us scientifically to understand their mechanism of action.
So, Patty, let's talk a little bit more about the partner with the beta-lactam. I've heard many people, very influential people, have said and pushed to pose this question: why don't you let clinicians, we have now a menu of options, choose the partner of beta-lactam? So you've been in the development of several of these drugs, and so how do people and pharmaceutical companies choose a partner? And maybe we'll discuss that concept later.
Right. So the choosing the right partner is, as Robert said, it's a good analogy of being like a good marriage. And over the years, the way that partners are chosen has changed somewhat, and the reasons why one would choose a certain partner has changed. Some of the original beta-lactamase inhibitor combinations were created by companies who had a beta-lactam, whose efficacy was now being diminished because of the presence of beta-lactams. And so the new BL-BLI combination were partnered to rescue that asset. For example, ticarcillin was partnered with clavulanate to make timetin, and piperacillin was partnered with tazobactam to make Zosyn, which, Zosyn especially has been a very successful way to rescue the asset that the company had of piperacillin. Certain beta-lactamase inhibitors are available orally, and clavulanate is the one that we have currently on the market. And so the only choices you would have to partner that with would be orally bioavailable beta-lactam antibiotics. Some of the newer beta-lactamases that are in the pipeline are also orally bioavailable. And so, again, you would be limiting your choices to those compounds that are orally bioavailable. Now, some of the newer, as I said, the really newer ones, such as the DBOs that we're going to be talking about a little bit later, you want to look at several things. You want to look at what are the organisms that you want to target. Is it Enterobacteriaceae? Is it Pseudomonas, Acinetobacter, all of the above? And can the compound actually synergize with the beta-lactamases that are in those organisms? Secondly, what's the spectrum of the inhibitor, and what organisms are those beta-lactamases found in? You also have to look at the pharmacokinetics of the inhibitor, and whether the pharmacokinetics match up with the beta-lactam partner that you want to choose. So there are many considerations for why you would choose a certain partner. Main ones being spectrum of activity, what antibiotic are you trying to rescue, and in which organisms. Those are the main factors.
So thank you very much. This is very useful. Robert, you're being very vocal about this in terms of not giving the free way to people to choose, and people try to do it because, one of the reasons, I guess, this knowledge is very specialized and not many people understand. So it's going to be very difficult that everybody will have the same level of knowledge. But there are other reasons that you are probably against trying to give leeway to people to choose drugs. Can you explain some of those?
Well, theoretically, you can do it. It's a very nice idea to have precision medicine. I'm all about precision medicine. And I think there are circumstances where you would want to use a cephalosporin over a carbapenem, or there are certain circumstances where you would want to use a particular type of cephalosporin over a particular. So I think, in a perfect world, to quote Samuel Johnson, that it's very attractive to be able to pair a particular beta-lactam with a particular beta-lactamase inhibitor, get the most out of the combination. What I worry about is that how you're going to implement this. How many different beta-lactams are you going to allow clinicians to pair with different beta-lactamase inhibitors? And what challenge will that be to the clinical microbiology lab to get that sort of data to support the clinician? What challenge will be there, for the, for the PharmDs to be able to, assist in the formulation of that particular combination. And you got to remember, the pharmaceutical industry spent a lot of time picking the right partner. It was based on very good science, based on PK/PD activity, based on microbiologic spectrum. And when we're given that choice at the bedside, we won't have necessarily all those things thought out. So unless it's a really careful decision, unless it's something that's based with support from the clinical microbiology lab and from the pharmacologist and with an understanding of the particular patient in mind, that's sort of, for want of a better term, precision microbiology is going to be challenging. Not that it can't be done. It can be done, but I think to do this in certain hospitals on the weekends may be very challenging.
Yeah, I couldn't agree more. I remind me with all these compounds that are coming out that I think is a very good news. I'm remembering what is happening with HIV and how these new drugs coming that you probably have a genotype to help you choose the drug, and based on that drug, I think this is going to be interesting as we move forward. But let's get back a little bit, Patty. What you probably were involved in the development of sulbactam and clavulanic acid and all that. So tell us about these first generation beta-lactamase inhibitors. I mean, how
Sure.
they were thought, developed, and no.
Sometimes it's hard to think back that far, but both sulbactam and clavulanate were developed when TEM1 was our problem. That was our one and only problem. Maybe some SHV1 thrown in on the side. But TEM1 was a very big problem in Haemophilus influenza and E. coli. And so, amoxiclav was developed really for upper respiratory infections because of the problem with TEM1 in Haemophilus. And like I said, sulbactam was partnered with ampicillin. It's IV only. Ampicillin was the workhorse for, things like, preoperative intra-abdominal surgeries, et cetera. And because of these beta-lactamases, it was starting to become ineffective. And so sulbactam rescued the efficacy of ampicillin. And after, maybe a decade's worth of use, maybe not even that much, we started to see, and also because of the third generation cephalosporins that were starting to be used, we started to see ESBLs. And from there, the world of beta-lactamases has just exploded. The first ESBL was reported in 1982, I think. And it was really a global problem. Late 80s. But so by that time, the need for new beta-lactamase inhibitors was really evident, and so tazobactam was developed then with the first ESBLs in mind, and with the first ESBLs, we're talking about derivatives of TEM and SHV, and that was the world that tazobactam was developed in.
And I was going to say one thing, Cesar. Patty alluded to some really interesting history, because, what's exciting about that time is that we didn't realize it. Now we know is that at the same time that the ESBLs were being discovered is all the same time the HIV epidemic was taking off. Not that the two are related, but, 1982-83 was a critical time in infectious diseases and the emergence of these ESBLs, first in the SHV family, and then in TEM, people couldn't put their head around it, like, what is this? And it's, it's a very interesting history. It's a very interesting history.
So, and I remember, outside of the United States, actually, sulbactam was paired with other drugs. I remember that combination of cefoperazone, sulbactam being used outside of the United States and mostly. But interestingly, I remember
it's still used in Asia.
Yes, correct.
Cesar.
And exactly, and I had the opportunity to visit some of my colleagues outside of the United States, in the United Kingdom, where resistance, the way we see it in the United States, is not as bad, and amoxicillin clavulanate and all that first generation inhibitors are still widely used as a first choice therapy. And it was for me shocking after being trained in this country, in the United States, being how different the epidemiology and the resistance pattern are, where drugs work or don't work. So, Robert, how about pip-tazo? Tell us about piperacillin tazobactam and how will be the next generation.
I became aware of pip-tazo because of the relationship of the Cleveland VA to Wyatt Ayers, I think it was called at the time. And there was a lot of clinical trials that started coming down the road. They wanted to use piperacillin tazobactam in the treatment of Pseudomonas aeruginosa. And, Cleveland got started with that. But then on a scientific basis, tazobactam, really was sulbactam on steroids. They're both sulfone beta-lactamase inhibitors. Sulbactam, when you compare sulbactam to tazo, tazo is a premier inhibitor. Tazo, very potent, very low turnover, very high affinity for the active site. It, has special relationship with piperacillin when it comes to the renal tubule and in terms of excretion outside. So tazobactam, was a step up. It was a real step up. And then used with a compound that is very potent, piperacillin. Piperacillin was still a very potent compound in terms of its anti-pseudomonal activity. Tazo didn't bring anything to piperacillin in terms of Pseudomonas. What tazo did is that it saved piperacillin from the other beta-lactamases that were around, and so that you could use a drug that had anti-pseudomonal activity and a little bit of anti-anaerobic, a good amount of anti-anaerobic activity. So tazo saved the day. It's interesting that sulbactam and tazo are pharmacologically derived drugs. Clavulanic acid is a natural product. So there kind of is a different philosophy there.
Yeah. So talking about piperacillin, although you say it is potent, as the organisms, particularly the Pseudomonas, evolved, clearly became weaker and weaker. And then we had this drug called ceftolozane that was paired with tazobactam. Patty, can you tell us something about that drug and why is one of the drugs that we are kind of using with that sort of what I call second generation beta-lactamase inhibitor, that tazobactam?
Yeah, so that's a very interesting combination. Ceftolozane is very potent in and of itself against Pseudomonas. Probably because it has really great entry into Pseudomonas aeruginosa. And as Robert mentioned, tazobactam does not potentiate the activity of anything really with Pseudomonas aeruginosa because of the AMP-C beta-lactamase. So ceftolozane is very potent against Pseudomonas. The tazobactam helps ceftolozane with ESBLs in Enterobacterialis.
And we may comment this, but now there are sort of new drugs being modeled for tazobactam. There is one that is being developed, and metazobactam, that seems to be a little bit more potent. Do you have any comments about that?
Yes, that one is, it is more potent than tazobactam. It is being partnered with cefepime, and it is meant for use with ESBL producing Enterobacterialis. We said that pip-tazo was designed really about the same time that ESBL started, but CTXM had not really hit the world yet. And so piptazo has lost some potency because of CTXM, but also because there are multiple beta-lactamases in strains. And so Tazobactam does help with some of those strains that have either multiple beta-lactamases or some of the CTX variants.
Okay, so that's a very interesting compound, which I'd say is second A generation. But then it came now the carbapenemases, right?
Well, before we go there, Tazobactam also does something very interesting. Like, even though it's linked to Tazobactam, one of the things that Tazobactam does is that it forms a unique cross-link across the active site, which the Tazobactam, to our knowledge, doesn't do. So it, and it also, because of the extra methyl group it may have, it has a little bit of a better activity than Tazo, I think. So, it's an interesting compound. I'm sorry to interrupt.
No. Fantastic comment. Thank you very much for that. So I am trying to think about in generation. People like to talk about generation in anti-infectives for some reason. I'm not a fan of that, but I think that then after the SBLs came the carbapenemases, right? With all this variety of different activity classes, etc. And I guess, one of the important first response was to get new beta-lactamase inhibitors that could inhibit those carbapenemases. And that I call that like the third generation. But these were chemically different and they were really interesting. And I always have problems trying to pronounce the bicycle octanes or DBOs. So, Patty, why don't you tell us this DBO like avibactam and relebactam?
Yeah, we will just say DBO. I think everybody prefers that. So avibactam was the first of the DBOs to be discovered and developed. It is not a beta-lactam molecule, but structurally it mimics a beta-lactam. So it really serves the exact same purpose, that inhibits the beta-lactamases. Avibactam, unlike the previous beta-lactamase inhibitors, is not turned over by the beta-lactamase. It is covalently bound, and so it's permanently stuck to the beta-lactamase, and there's no turnover. There's no turnover of the beta-lactamase. So these are very potent inhibitors. Avibactam has a broad spectrum of inhibition. It inhibits class A enzymes, really all of the class A's quite well. It inhibits class C. It was the first inhibitor to come along that inhibited the AMP-C beta-lactamases that are found in Pseudomonas and some species of Enterorectalis, and it inhibits some of the class D enzymes, including OXA48, which is an important carbapenemase. So this was really the first major step forward in some of the beta-lactamase inhibitors. The other DBO that is on the market already is relebactam, that is partnered with imipenem. And that one has a different spectrum of activity because it is partnered with imipenem. The actual enzyme activity isn't all that much different than avibactam but because it's partnered with imipenem, it covers a different space. But there are numerous other DBOs that are in the pipeline: zidibactam, nacibactam, et cetera, et cetera. So there's several others in the pipeline.
So, Robert, can you comment about OXA48 in avibactam versus relebactam? Because this is a question that a lot of people tend to ask in terms of it. And the spectrum of activity, as Patty mentioned, imipenem relebactam, I'm thinking about a lot of Pseudomonas, where the ceftazidime avibactam more directed to sort of a carbapenemase producers, Enterobacterales.
Well, it's interesting. OXA48 is, we don't see too much of that in the U.S. To be honest with you, we see a lot of KPC, but our our colleagues in Europe see a significant amount of OXA48. So, the dividing line is the water. And, you have compounds like, ceftaz-avi, and, OXA48 being a OXA-type enzyme can be inhibited well by the avibactam. The other thing is that the OXA48 doesn't do a good job taking out ceftazidime. So when you use Taz-Avi, you're giving OXA48 bearing cells like a 1-2 punch. They don't do a good job with the cephalosporin. They get inhibited by the avibactam, and it's an important combination to consider in the treatment of patients with that. Unfortunately, we didn't mention it yet, Weber-Bactam doesn't do a good job with OXA48. So when you have a carbapenemase, Marrow-Weber may not be a good choice. Imirel, I think I prefer Taz-Avi in this particular situation because I like to use the cephalosporin component of it in addition to the avibactam component. I'm always a little bit, I don't know that relibactam is at least hasn't been clinically my first choice against OXA48. I've kind of stuck with Taz-Avi.
Okay.
And a lot of hospitals don't have Imirel on formulary, so I don't know why. It's an important drug to have. It does have potent activity, but
Yeah. Any comments, Patty, on the pseudomonal activity? Because we've used imipenem relebactam, particularly when the ceftolozane tazobactam shortage appeared. That was one of our anti-pseudomonal options for many reasons. And you see most people and literature suggest that it has good activity against pseudomonas, and reserving ceftazidime avibactam, and we'll talk about in a minute, to meropenem verbactam for more the enterobacterialis. Any comment, Patty?
No, Robert was correct. I think the main take home for imipenem relebactam is that it, as he said, it is not good against OXA48. So if you do suspect that it is a carbapenemase, then you should not use that.
Okay.
But it is good against pseudomonas, you're right.
Correct.
On this point,
sure.
I mean to interrupt, Cesar, but interestingly, if you have these variants of KPC that emerge in the clinic that are resistant to Taz-Avi, Imirel is a very good choice because those enzymes like D179Y can't break down imipenem. They become more imipenem susceptible. The relibactam gets in okay against those KPCs. Relibactam's got great activity against KPC. And Imirel would be a good pharmacologic and biochemical and microbiologic choice against Taz-Avi resistant variants that emerge because of those mutations that give you Taz-Avi resistance. So, you got to weigh both sides.
Yeah, and that brings me to now talk about Weber-Bactam, Patty, which is different chemical class, interesting chemistry. So boronic acid derivatives, but we much more targeted a spectrum, and now partner clinically with meropenem. Any comments?
Yes, I can tell you a brief bit. Robert might be the better one to really expand on it. So as you mentioned, Weber-Bactam is a boronic acid. It was specifically designed to work against KPC, and it has really very good activity against KPC, and it has activity against some of the KPC variants that we have seen that become resistant to avibactam. It does not have any activity against OXA48, and which is readily hydrolyzes meropenem. So that combination is still a problem with OXA48. But Robert, would you care to comment any more about the use of meropenem Weber-Bactam?
I like to think of meropenem Weber-Bactam as the first step. And the reason I say that is that the DBOs are really good compounds. Weber-Bactam, in principle, is a very good beta-lactamase inhibitor, particularly against, KPC and Class A and all that. And, there's got this gap against, it's got a gap against OXA48. But as an idea, the boronates are a really good idea. What I think Weber-Bactam's secondary contribution is that it allowed people to explore the other boronic acid space. So the new compounds like taniborbactam that are coming down the pike and QPX, they're like, sons and daughters of Weber-Bactam, but they go where Weber-Bactam has not been able to go. They're really broad spectrum inhibitors. So I think Weber-Bactam is like sulbactam in the boronic acid space, and Clav, they did a good job. And, Weber led the way to the development of other things, even more potent boronates, in my opinion.
And just to go to the comment about resistance, so I do prefer, when I have certainty that I'm treating a KPC, to use this compound because of this variety of reasons, particularly the evolving resistance to ceftazidime-avibactam and the hypersusceptibility to the carbapenems when that occurs. Ryan Shield was here last week, presented very interesting data when that happens, maybe with other beta-lactamase inhibitors. So I think that's a very important point to discuss, and we will touch upon the resistance later in the podcast. But I want to move on now and talk about then certain others that have been I've been trying to develop against specific vacuums in the spectrum. And one of the vacuums that we have, even with these newer beta-lactamase inhibitors, is Acinetobacter, right? And there is one in the pipeline, hopefully soon available, called sulbactam durlobactam, which is, interestingly, two beta-lactamase inhibitors, but they do have other activities. Patty, can you illustrate that point?
So this is a really interesting combination. So durlobactam is a DBO type beta-lactamase inhibitor. It was part of a rational drug design program that wanted to expand the spectrum of activity against class Ds. So if you recall, avibactam, Class A, good. Class C, good. Some Class D. So the point of this program was really to expand on Class Ds. Well, where do we find a lot of Class Ds? Acinetobacter. And so the idea was to partner it with something that would really, target Acinetobacters. Interestingly, sulbactam, good old sulbactam from the 1980s, has this really unique property that it binds to the PBPs of Acinetobacters and Haemophilus and Neisseria. It's a very odd combination of organisms, but it inhibits the PBPs of those organisms. It used to be, back in the olden days when there was really not a lot of resistance, that people would use sulbactam for Acinetobacter infections because of the sulbactam activity. But, over the years that has diminished. So sulbactam durlobactam combination is utilizing sulbactam as the beta-lactam drug that is being rescued now by the inhibitor durlobactam. And it seems to have worked quite well. This is being developed by Therapeutics. They disclosed their phase 3 results last year. It seemed to have, really good efficacy. It's now at FDA, and as you said, hopefully it'll be available to patients soon.
Robert, any comment?
Well, durlobactam actually, to Patty's point, I'm glad it's a smart drug. Durlobactam is a smart drug. It's, Class D activity is very. It's also got some Class C activity. So, so when you're in Acinetobacter, if you want an inhibitor to inhibit Class D and Class C, durlobactam is it. It also has some. Interestingly, it has some PBB activity, but it affects the morphology of cells, but it doesn't affect the MIC. So Dorolobactam is binding there, but it's not bringing the MIC down. It's when it's combined with sulbactam, and Patty mentioned, the PBB activity of sulbactam, that the two drugs work extremely well together. And, it's it's a real interesting, notion that, we've been trying to develop. It's like, target redundancy or multiple target inactivation. And the compound, like, ceftazidime inhibits a PBB and avibactam inhibits, the beta-lactamase. You have a sulbactam inhibits the PBB, and maybe there's some other enzyme that inhibits the rolobactam, inhibits the beta-lactamase. Maybe it inactivates some of the cell wall things. So, I think as you get at more targets against these multi-drug resistant organisms, you see the benefit of the combination. But I think we still have to see the formal results of that trial published. To my knowledge, the final publication of that trial has not been out there, and the FDA is evaluating it.
Yeah. Okay. And the next vacuum in this generation that we've been reviewing now is the famous metalloenzymes or metallo-beta-lactamases, right? That are very problematic, particularly outside of the United States, but we are seeing more and more here in the United States. And none of the ones that we've discussed so far really had a good activity. And you can throw there Stenotrophomonas maltophilia, which has an inherent metalloenzyme as well. And using a combination, for example, of a beta-lactam compound like aztreonam, which is the only one that we know has some activity against metallo, and combining with ceftazidime avibactam, we've used that for these NDMs or other metalloenzymes in clinical practice. But then there is a possibility of this developing of the compound called aztreonam and avibactam. Robert, can you comment on this metalloenzyme inhibition?
That's truth be told, Professor Alejandro Villa a long time ago showed that in looking at another metallo-beta-lactamase is that aztreonam could not form a productive complex with the metalloenzyme. So that's why if you have some strains that have a metallo-beta-lactamase and don't have other enzymes that can break down aztreonam, you will see susceptibility to aztreonam alone in some strains of Pseudomonas. So I think this obligates us in the clinical microbiology lab when we take care of patients to make sure that the clinical lab, if there's a metallo there, at least testing aztreonam alone. So why did the combination of Taz-Avi and aztreonam get proposed? And how is it different or the same than aztreonam avibactam? Simply put, at the time that Taz-Avi aztreonam combination was explored, aztreonam avibactam was not on the market. It was only in development at that time. There was concern, historically speaking, that you always got to be a little worried about high dose aztreonam. There's some diffuse hepatic injury that happens. It's reversible, but aztreonam is a great drug, but makes some ID docs a little skittish, particularly when the LFTs go up. But what the Taz-Avi aztreonam combination did, and this was very nice work that was championed by Dr. Maria Mojica at Case Western Reserve University, where she was able to show that by using the avibactam part, you inactivate many beta-lactamases of the class A and class C variety, let's say like L2, and you inactivate that. Using the aztreonam part, you bypass those enzymes that break down aztreonam, and since the metallo can't break it down, it bypasses the metallo. Ceftazidime acts to hit multiple PBBs and also probably acts as a decoy, to keep the other enzymes busy so aztreonam could reach the penicillin binding. This is speculative. This is a hypothesis. But nevertheless, Maria Mojica did very nice work to show this. Aztreonam avi, aztreonam avi, here you have a combination The Acinetobacter is still bypassing, right? The avibactam is still protecting the Acinetobacter. But the question remains is that, do you gain any benefit from the extra beta-lactam? Do you gain any benefit from it? And, the trials need to be done. Truth needs to be seen. But the most important thing is that those combinations of a monobactam or a cephalosporin, with a monobactam that bypasses and inhibiting the serine enzymes in these other metallo-beta-lactamase producing strain is a strategy that I think we're going to welcome in the clinic against metallos. The problem also arises as, do you believe that those metalloenzymes actually have a clinical impact? And work that has come from Professor Nicolau's lab raises some things into question about the potency of the metalloenzyme, the role of the metalloenzyme in these infections. So, unfortunately, it's more complex.
Yeah, Patty, the talking about the metalloenzymes, then we have this newer generation of more now broad spectrum, ultra broad spectrum beta-lactamase inhibitors that are going to hit everything, right? And there are some in the market, like taniborbactam. You talk about Cesar and other compounds. So what do you think about those, and how likely are to get to the finish line?
So let's start with the DBOs, Zidibactam and Nacubactam. Those are both still in development. Those as beta-lactamase inhibitors, they do not inhibit the metallos, but they do have intrinsic activity against some of the species of Enterobacteriaceae. So you will see activity of the combinations against certain metallo-beta-lactamase producing strains because of the intrinsic activity of those two inhibitors against the E. coli, Klebsiella, et cetera. Now, taniborbactam is an interesting compound. That's the next generation of boronic acid inhibitors, as Robert was talking about earlier. That has a very broad spectrum of activity against class A, class C, and now the metallo-beta-lactamases. For the metallo-beta-lactamases, there are three main groups of enzymes: NDM, IMI, and VIM are the three main classes of metallos. Now, taniborbactam inhibits NDM very well. It inhibits VIM very well. It does not inhibit the IMI enzymes at all. The IMP, sorry, not IMI, the IMPs. So if you're in the United States where NDM is prevalent, great. If you're in Europe where VIM is very prevalent, great. If you're in Asia where IMP is the most common enzyme, it will not work. But it is the most promising, the most broad spectrum that is the latest in development. That drug is being partnered with Cefepime. They have finished their phase 3 trials, and I don't know where they are in their submission process, but they announced the results of their phase 3 trials sometime in the summer, I think.
At ID Week, I think they announced they had a session where they and it did well in the phase 3 trial.
Yep. And there's another inhibitor being developed by QPEX, which is extremely broad spectrum that does include the IMP beta-lactamases. So that one is much, much earlier in development, but when that one comes along, that would cover all of the classes that we know about currently.
Robert, any comments?
No, these are smart drugs, and there's been some very nice crystallographic work explaining why they work, and they really advance as boronic acid inhibitors both advance the spectrum of activity and get into the metallo world. With everything in ID and with everything in medicine, you always you always have to be cautious. Things will happen. We don't know what they are yet, but just like people have predicted pandemics, it's likely that resistance to these agents are going to rise too. And but I don't want to be a naysayer. I like Cidobactam. I like its activity. It's got intrinsic. I think NACU is also a very good drug. Cidobactam does beta-lactam enhancer. We've worked with it and been very, very pleased with its activity, especially against Pseudomonas. So, I really like those drugs. I think they're good additions. But I think one of the key things, and I saw in a nice meta-analysis that was published recently, is that these new agents, they're not going to buy you real estate against the easy to treat things. The easy to treat things are still going to be easy to treat with that. It's only in these specialized circumstances where these agents are really going to make a difference. And I think we need to be conscious of that, conscious of the stewardship issues, conscious of driving resistance even greater. So I think when these very important and powerful drugs get released, we have strategies in mind, to implement their, to help their implementation in the clinical arena. So I think I welcome these drugs, but I think we're going to have to use them very judiciously. And I think the hard part for hospital formularies is going to be, which one do you pick? You can't pick all 5. You can't pick all 12. It's economically unfeasible. So I think, maybe ASM's job is to provide guidelines, for people to do this, or help the clinicians and help the clinical microbiologists, with a set of, these are the better agents for and make it very clear. These are the better agents for these targets. And, because of clinical trial evidence, it's not just biochemistry or microbiology. You got to have clinical trials.
Yeah. And I'm going back to my analogy with HIV. Having this many of drugs is going to imply that you need to really know what you're treating, So, Patty, can you comment about the importance of molecular diagnostics and personalized diagnostics? Because that will be the only way in which you eventually can treat things.
Yes. So that's a great question. I can comment that technology exists for, it's not bedside diagnostics by any means, but it is, within a half a day, diagnostics for certain pathogens and certain resistance mechanisms. For example, we talked about Sulbactam durlobactam earlier. It's only good against Acinetobacter. And so you have to know that your patient has an Acinetobacter to give the drug, right? So there are platforms available where within, like I said, 4 hours or something, you can take BAL fluid and you have an identification of, does it have Acinetobacter, yes or no? That kind of thing. Those exist. There are platforms that you can have not from, from some bodily fluids, mostly from a culture plate, but you can identify, does it have a carbapenemase and is it KPC, oxa, or a metallo, yes or no? I mean, those are the kinds of things that exist today. So, but those platforms are pretty sophisticated and, it requires an investment on the hospital's side to have those instruments. Now, one could argue with antimicrobial stewardship that it would pay off in the end for you to actually know what you're dealing with and have correct treatment right up front. So those are the kinds of things I think that we'll have to consider moving forward, is that, I think the days of having, one size fits all empiric therapy that we use for everything, those days are gone. And so I think that going forward, we really do have to focus more on what's possible in microbiology, and make the investments both in further development, but also at a hospital level to make the investment to have those available.
Very, very wise words. Thank you very much. Robert, I'm going to give you the last word briefly. And what's the future of this? Put your crystal ball and tell us what's going to happen with all these drugs and in the near future on this?
Well, like one of our, it's always hard to predict the future when you're living in the past, but nevertheless, history will repeat itself. History will repeat itself. We know that. We know that evolution is a strong driving force in the world. And, I think these drugs will find a place. I think eventually, the combination of wise stewardship and rapid molecular diagnostics and all the advances we're getting with therapeutic drug monitoring are going to help our patients. I also think that, we're all of the generation where we know that there's going to be something added to antibiotics down the road. Like, we're not just going to be giving patients an antibiotic or a beta-lactam, beta-lactamase inhibitor. We're going to be using immunomodulators. We're going to be using, antibodies. We're going to be using phage therapy. The situation is going to get much more complex in some ways, whether we like it or not. But for these compounds, I think the future is bright. I think we're just going to have to figure out how to use them best. And that will take time. That will take time and money.
Yeah. So that means your job security for infectious diseases. So infectious disease is the best branch of medicine. So this just tells you how exciting the future is, how complicated, but that is a challenge that we all welcome. So I want to thank you both for this fascinating conversation. I have learned a lot, as usual, is in these shows with experts in the field like you. I hope you also learned something today. I am Cesar Arias, Editor-in-Chief of AAC, signing off.
Thank you.